Hello and welcome everyone. Thank you for joining today's webinar hosted by Gen Pharmatech. My name is Koming and I'll be your moderator for this session. Today, we are diving into a critical and very timely topic, how to navigate the evolving landscape of cancer immunotherapy in the post-PD-1 era. Immune checkpoint inhibitors, particularly those targeting PD-1, have really reshaped cancer treatment, offering durable responses for patients who previously had limited options. However, as these therapies become more widely used, we are increasingly confronted with the realities of primary and acquired resistance. And this highlights the urgent need for next-generation strategies that go beyond monotherapy. So in this session, we'll explore the current challenges and resistant mechanisms in anti-PD-1 therapy, the emergence of combinatorial approaches such as anti-CTLA-4 antibodies and ADCs, and the importance of robust predictive preclinical models that can better mirror the immune-tumor interactions driving treatment response or failure. So whether you're working in oncology drug development, immunology, or translational research, today's content will offer valuable insights into designing and validating therapies in this complex therapeutic space. Now, before I bring on our speaker today, I want to briefly introduce our company, Gen Pharmatech. Gen Pharmatech is a global provider of integrated preclinical services built on the world's largest library of genetically engineered mouse strains. Our capacity allows us to conduct more than 1,000 preclinical studies per year, and our expertise covers a wide range of disease indications from oncology, immuno-oncology, neurodegenerative diseases, vaccine development, and beyond. This comprehensive suite of preclinical service, backed by our more than 25,000 GEMs and advanced preclinical platforms. Supported by a team of 1,500 scientific experts, our infrastructure spans 7 subsidiaries across China and the US, with large-scale SPF, vivarium capacity, and over 6,000 new models generated annually. Whether you need breeding, custom model design, or full in vivo studies, we provide integrated scalable support. Our newly expanded capabilities in the US, our San Diego site, position us to partner more directly with biopharma teams in the North America that are navigating early discovery through IND-enabling work. Now it's my pleasure to introduce our speaker for today, Dr. Jun Lee, who is our Head of Oncology Pipeline and Gen Pharmatech. Dr. Lee has deep expertise in preclinical model development for oncology and immuno-oncology and has worked closely with research teams across biotech and pharma to support the advancement of novel cancer immunotherapies. Welcome, Dr. Lee, and please take it away.
Thank you, Kaming, and good morning, everyone. This is Jun from Nanjing, China, the headquarters of Gen Pharmatech. So today I'm going to talk about the post-PD-1 era in cancer immunotherapy from these 3 aspects. For the first part, I'm going to talk about the challenges and the resistance in the anti-PD-1 therapy. So as we know that the PD-1 therapy has been popular for decades, but it also face serious challenges nowadays. The main challenges in the PD-1 therapy I have listed here, summarized. So the first is the resistance. So either primary resistance or acquired resistance, which can be caused by the loss of antigen presentation, for example, or the alternative immune checkpoint upregulation, or there's defects in the JAK, and gamma pathway. Also, it can be caused by the changes of the microenvironment become immunosuppressive. And the second factor that can contribute to the PD-1 resistance is the we know that the tumor can be defined as either cold tumor or hot tumor. So for the cold tumor, which means there's a low tumor infiltrating lymphocytes, so there's less T cells infiltrating into the tumor. Either it could be caused by the physical barrier, or there's a vascular barrier, so the T cells cannot penetrate into the tumor in the center, or it can be caused by the lack of neoantigen to present it by the antigen presenting cells, such as the DC cells or macrophages. So the T cell cannot be activated, or it could be caused by the suppressive stroma around the tumor and prevent the T cell to enter into the tumor. And the third reason could be the caused by the myeloid immunosuppressive compartments or the stroma, which we know that the MDSC or the tumor-associated macrophages can blunt the T cell, NK cell activity. And the fourth factor can contribute to the PD-1 resistance or cannot be, I mean, the doses is limited because if you use a combination therapy, it can raise the severity or the adverse events. It's called immune-related adverse events or toxicity in the organs specific. Some is like, on-target off-tumor toxicity caused by the anti-PD-1 or combination with PD-1 therapy. So this will limit the dosage in the clinic, which contribute to the lower efficacy. So the management of combination therapy in the clinic is not perfect, so it can blunt the anti-tumor immunity. And also, the other factors contribute to the challenges includes the lack of predictive biomarkers or patient heterogeneity. So such as the PD-L1 as a marker, if we recruit the patients from, we test the PD-1 expression in the patients, and we try to recruit the high PD-1 expression into the clinical trials, or the tumor mutational burden as another marker to recruit patients. But these markers are not perfect. So also the inter-patient or the intra-tumor heterogeneity complicated the patient selection for the clinical trials. The other one is the translational gap. We always try to find the suitable preclinical models which can predict the human responses, but there's limitations such as the species differences or the donor variability for like humanized mice. And also the limited myeloid reconstitution in the humanized mice reduces the predictive power in the clinics. Also, as you may heard that last year in December, Merck has announced announcement saying that Merck discontinued the combination therapy of PD-1 or Keytruda combined with anti-TIGIT antibody and the anti-LAG3 antibody. It clearly shows that even in theory, even in theory, we know that the two pathways combined together should have a better efficacy than monotherapy alone. But clinical trials shows that the combination therapy failed clinically, which indicated that the mechanistic rationale alone is not sufficient. So we talked about the main challenges, but in terms of the strategy, how should we overcome those challenges? So here, at least I summarized from the literatures that the first, we need to overcome the resistance or the cold tumors. So we turn to the cold tumor into the hot tumor. Either we can use the intratumoral STING or toll-like receptor agonist, or we use the oncolytic viruses, or like local radiation. And we can also try to like fix the angiogenesis to make the blood vessels normalized, which can help the T cells to infiltrate. Also, we can increase the antigen genicity, and such as we use neoantigen vaccines or epigenetic modulators to increase the MHC antigen expression. In terms of the antibody structure, nowadays, there's quite a lot in the preclinical trials that people design the bispecific antibodies or the T cell engagers. And this type of antibody or antibody engineering showed quite promising results in the preclinical models. And the other strategy is to target the suppressive myeloid or stroma cells, like the inhibitors of the CSF1 receptor, the CCR2 inhibitors, or TGF-beta inhibitors, or we try to program the tumor-associated macrophages, or we can combine the myeloid targeting agents with the PD-1 blockade. Because inside the tumor, especially the cold tumor, there's quite high number of myeloid cells resident in the tumor, which makes the tumor microenvironment quite immunosuppressive. And the other part to use is cell therapy, like the CAR T therapy or the TCR-T therapy. Also nowadays we have CAR NK therapy or CAR macrophage therapy can be used or combined with the PD-1 blockade. And also the next generation cell engineering addresses like trafficking or T-cell exhaustion, or turn the hostile tumor microenvironment into friendly microenvironment. And as I just mentioned, if we instead of monotherapy, we can do the antibody engineering to change the structure of the antibody, become like bispecific antibody. So one arm targeted CD3 T cells and the other arm targeted the TAA. Can also have more efficacy compared to the PD-1 alone. Also, we can modulate the systemic factors such as microbiome modulation or the metabolism of the tumor, or the cytokine engineering such as IL-2 or IL-15, which is critical cytokines for T cell, NK cell proliferation, to reshape the systemic immunity without causing too much toxicity. So the second part, I'm going to talk about the preclinical models for combination approaches with anti-PD-1. So here is just some examples showing you the combination of Keytruda, for example, the graph on the left, with CTLA-4 antibody, the ipilimumab combined together in the CD26 tumor expressing human PD-L1. So as you can see that the red line is the Keytruda, the blue line is the anti-CTLA-4 antibody alone. But if you combine these two together, it definitely shows a synergistic anti-tumor effects. Also in this B16F10 melanoma model, in this B6 mice, which express human PD-1 and human LAG-3. So we combine the PD-1 antibody with anti-LAG-3 antibody together. As you can see that it definitely delayed the tumor growth compared to the PD-1 or anti-LAG-3 alone. And it's also prolonged the animal survival by the combination therapy in this animal model. Another example is to combine the Enhertu with Keytruda. So this is the MC38 mouse colon tumor expressing human HER2. We inoculate the cell line into the B6 mice expressing human PD-1. As you can see, the combination therapy, which is the line, the blue line at the bottom, it led to the complete tumor regression from day 10 until day 28. And they definitely have a longer survival of the animals in the combination therapy of Keytruda with Enhertu. In addition to the models I mentioned, we at Gem Pharmatech have a number of different mouse models can demonstrate the concept that combination therapy definitely works in these animal models to test the antibody you are interested, such as the PD-1 combined with GITR, PD-1 combined with VISTA, or PD-L1 combined with CD28, or PD-L1 combined antibody combined with B7H3 in different animal models. They all shows synergistic effects. So except these models, we also have the NCG mice, which is immunodeficient. And these mice don't have the TB or NK cells. They don't have TB and NK cells. They only have reduced functional macrophages and DC cells. So in the US, the Charles River is exclusively distribute our NCG mice in Europe and the US. And this mice has been used quite widely in the publications, as you can see. So instead of the syngeneic models, this is the human immune system reconstituting model. We call the human PBMC NCG mice. So we use human PBMC inoculated into the NCG mice, and it can create a human immune system in this immunodeficient mice. Here is just the in the blood, we tested the human CD45 cells, especially the CD3 is the main subtype in the mouse blood after the reconstitution. As you can see, the CD3 positive cells can last from week 2 to week 8. Also, the CD8 positive T cells and the CD4 positive cells can have a higher percentage from week 2 to week 8. So to test the human PBMC reconstituted NCG mice model, we can give you an example by using the immunocheckpoint inhibitors. So here are two PBMC donors, the PBMC donor 1 and the PBMC donor 2. We use the anti-PD-L1 antibody, the Tecentriq, and inoculated the mice with MDMB-231, which is triple negative breast cancer. So you can see that the the donor 1 is the red line, and the red line is the PBS, and the blue line is the treatment group with Tecentriq at 20 milligram per kilo. It has 45% of tumor growth inhibition. Another donor is the donor number 2, and the control group is the green line, which is similar as the donor number 1. After you treat it with the Tecentriq at a frequency of twice a week for 3 times, for 3 weeks, it has generated the anti-tumor efficacy about 56% of the tumor growth inhibition. Another example is we use the RKO cell line, which is quite poorly differentiated colon carcinoma. So the blue, the black line is the control of the NCG PBMC reconstituted model mice, and the treatment shows the 45% Tecentriq efficacy. Instead of the monotherapy and the combinational therapy, we also have mouse models can be used to test the bispecific or the T-cell engager. So here is the example of the AK112, which is a bispecific antibody of anti-PD-1 and anti-VEGF. So we compared the bispecific with each of the arm alone, the Opdivo anti-PD-1, and this bevacizumab with anti-VEGF monoclonal antibody. So you can see that the bispecific has better anti-tumor efficacy in this PBMC NCG tumor model. This graph just shows that in the LS174T, the human colon cancer inoculated in this model, we have used the T-cell engager, which is anti-CEA and the CD3 T-cell engager compared to anti-PD-1 and anti-PD-L1 monotherapy, and we combine these two together. So the bottom two lines, the green and the purple line, shows that the combination therapy of the immunocheckpoint inhibitor of PD-1, PD-L1 plus the T-cell engager has definitely shows a stronger anti-tumor efficacy. The third part, I'm going to talk about the preclinical mouse models for efficacy and the translational research. So here I listed in a table that the types of mouse models we have. So the first one is the synergistic immunocompetent model that I just showed you in the previous slides. And we have the human PD-1, PD-L1 knock-in mice, which means these mice express human PD-1 and PD-L1. And also we have the human PBMC reconstituted in the NCG mice model, and we call it humanized mice. Also, we have the human stem cell reconstituted NCG mice model. And we can also have provide the service on the PDX with NCG humanized mice. And lastly, we have this genetically engineered mouse model, which means this after the certain genes has been modified, such as KRAS or EGF, these mice can spontaneously generate tumor. So each model has its application area, right? So they can be used under different scenarios. For example, if you look for the mechanism of T-cell myeloid interaction, you can choose the syngeneic immunocompetent mouse model because it has the intact immune system. If you're testing the PD-1, PD-L1 in an immunocompetent setting, you can consider to choose the PD-1, PD-L1 knock-in mice. Also, if you look for a short-term proof of concept when the human T cells is required, you can choose the human PBMC humanized mice. For longer term human immune system reconstitution, you can choose or consider the human stem cell humanized mice model. If you are interested in the personalized tumor architecture with human immune response, the PDX model may be like can provide a better microtumor environment because the PDX is from the human tumor. The human tumor compared to the mice, compared to the tumors, mouse cell lines, it has its heterogeneity. It's more like it's kind of mimic the real like human tumor in situ. So if you want to test like ADC or neoantigen vaccines or the combination therapy in a real human tumor, PDX could be a better choice. But each model has its pros and cons, right? So such as the human PBMC humanized mice, that because it will lead to the GVHD, so it has a short therapeutic window. But for the human stem cell reconstituted model, it has multi-lineage human immunity. It not only have the CD3 T cells, but it also have some of the myeloid cells as well. So I'm not going to go through each detail of each model. If you are interested, one of those models you can contact contact us after this webinar. We also have these resistant cell lines. For example, the PD-1 resistant CT26 PD-1 resistant cell line. So we inoculate the tumor in the mice, and we gave the Keytruda treatment, and then we isolated the non-responsive tumor from the mice and dissociated and culture them in vitro, and then we repeat this process like 4 times, and then inoculate back to the mouse, and finally we got a CD26 PD-1 resistant cell line. And then here is the validation. And on the top graph is the wild type CD26, and the bottom graph is the CD26 PD-1 resistant cell line. As you can see that if you gave the tumor Keytruda at 10 milligram per kilo, basically it doesn't have any effects compared to the control. And compared to the wild type, it just shows the wild type has much better effects responsive to the Keytruda, but the resistant one, they all grow quite fast. And we also analyzed the tumor by flow cytometry and to look at the immune cell profiling in this resistant tumor. So the black bars represent the wild type, the red bars represent the the resistant cell line. As you can see that there's quite substantial reduction of the CD4 T cells, CD8 T cells in the tumor itself. Also, there's an increase of the MDSC in the resistant cells. That's why we, from the mechanistic point of view, that's why this tumor is resistant to Keytruda, because there's less T cells inside the tumor, but more like immunosuppressive myeloid cells in the tumor. We also have developed this CD26 B2M knockout cell line. We inoculate this B2M knockout cell line into this BARBC mice expressing human PD-1. As you can see that the solid line, the 2 solid lines, the black and the red, represent the knockout B2M knockout cells tumor. The red line is the treatment group with Keytruda, so it doesn't have much effect, only have like TGI about 15%, almost the same as the control group. So it definitely shows the resistance to PD-1 by knocking out the B2M. And compared to the wild type, so this is the wild type control group, and the dotted red line is the wild type treated with Keytruda. It has like 70% of TGI. We also analyzed the tumor microenvironment and tumor infiltrating lymphocytes by flow cytometry in this tumor. As you can see, when you knock out the B2M, there's no MHC expression in the resistant cell line, which means it cannot activate the CD8 positive T cells. That's why there's no efficacy. So in addition to those cell lines, we have also developed the STK11 deficiency cell line. One is the MC38 knockout STK11. One is the CT26 knockout the STK11. So we treated the tumor with anti-PD-1, anti-mouse PD-1. This is anti-mouse PD-1, the RPM 114. And we treated this with TNG260, which is a small molecule. The TNG260 is an inhibitor of the co-rest, which is specifically designed to reverse the anti-PD-1 resistance, because if there's a STK11 mutant in the cancer, it's resistant to the PD-1 therapy. So we combine these two together. As you can see that it definitely reversed the PD-1 resistance in the MC38 as TK11 knockout tumor. It also shows some efficacy when you combine this together with the CT26 knockout as TK11. And the monotherapy doesn't have any effects in this tumor model. So if your research is related to the STK11 deficiency, maybe this is the preclinical model you can choose. And on the other side, that we also have the syngeneic mouse models. The syngeneic mouse models are like resistant to PD-1 therapy in nature. For example, the PANC02, the B16F10 melanoma cell line, they are very cold tumors that don't respond to PD-1 therapy. Also, the LLC1, the MPC11, the 41, they have very limited efficacy response to PD-1 therapy. So if if you are interested in these syngeneic models resistant to PD-1, and you can contact us and look for the specific cell line. So at last, for the CDX models, we have a quite big variety of cell lines you can choose, from leukemia to lymphoma, from blood cancer, multiple myeloma, and the lung cancer, and non-small cell lung cancer, small cell lung cancer, liver cancer, cervical cancer, sarcoma. And there's a huge number of cell lines we have developed at the CDX model. And they can either use in the NCG mice, or they can be used in the human immune system reconstituted mice, the PBMC NCG or the HSC NCG, or even we have the NCG expressing human IL-15 as well. So if you are interested in any of the models, please do contact our BD team. Right. This is the last slide. Thank you very much. And if you have any questions, please, we can discuss. Thank you.
Yeah, thank you, Dr. Lee, for the insightful and really thought-provoking presentation. So we'll now proceed to the Q&A session, and we're opening the floor for any questions to be entered into the Q&A chat box. Please feel free to drop your questions again into the Q&A chat box or chat, and we'll get to as many as we can today. Okay, so we actually have the question come in during our presentation. So Dr. Lee, you mentioned several mechanisms of resistance to anti-PD-1 therapy, such as the B2M knockout and the STK11 deficiency. Can you expand a little bit on how these specific alterations would influence the tumor microenvironment, and what implications would they have for designing combination therapies?
Yes, thank you, Kamin. So for the B2M deficiency, because B2M, if you make it B2M knockout, it will lead to a loss of the MHC1 expression. But MHC1 expression is very important. It's crucial for the CD8 T cell activation. So this will make the cancer cells like invisible to the CD8 T cells. So even the CD8 T cells can be activated, but it can't recognize the cancer cells or kill them. So that's the B2M deficiency, why the B2M deficiency can cause resistance to PD-1. The STK11 is, the function of STK11 is to make the tumor microenvironment more like cold, more immunological cold. It reduces the T cell infiltration into the tumor and recruit more like immunosuppressive cells like the MDSC or the TAM. So in terms of mechanism, the B2M deficiency or STK11 deficiency will make the tumor microenvironment very unfriendly to the T cells That's why the T cells cannot kill them. So yeah, if
Oh, mhm.
Sorry, what's your second question? I
Oh,
forgot.
no, yeah, no, that was it. So I think that's great. And I think, it definitely speaks to the different roles that different molecules, such as, MHC1 and, STK11 play in modulating, the immune landscape in tumors. So, thank you very much. I think it's a very, very, very insightful. So the second question we've come in is that can you elaborate on the role of bispecific antibodies and T-cell engagers in overcoming resistance to PD-1 blockade? Yeah.
Yeah, so the bispecifics or the T-cell engager, so physically one arm binds to the CD3 T cells, the other arm binds to the TAA, which is the antigen expressing on the cancer cells. So the bispecific would physically bind these two kind of cells together, and forms kind of like a immune synapse. So the immune synapse will drag these two together, because it will like bypass the need of MHC1 presentation. So normally the T cell need to be activated by the MHC1 presentation of the new antigen. But if you use bispecifics or T cell engager, it will bypass this part. It will activate T cells like without this pathway, and because it drags the tumor cells next to it, so it will kill the tumor cells like immediately in its proximity. So that's why this type of antibody is quite popular nowadays compared to the monotherapy alone.
Thank you very much for that. So in the talk we've talked a little bit about some of the humanized mice like the PD-1 and the human PBMC, humanized NCG for testing checkpoint inhibitors. So for any researchers out there who are developing novel immunotherapies, how should they decide between choosing using a humanized model versus a syngeneic model?
So if your antibody is kind of like using the human CD3 as a part of your target of your antibody, because the human PBMC reconstituted mice, the main type of the immune cells is the CD3 T cells. So for example, if you design T-cell engager, I think the human PBMC reconstituted NCG is a better model for you to test your T-cell engager. But the disadvantage is it has a short therapeutic window. It will lead to the GVHD in the animals, and the immune system in the mice is not intact. It doesn't have much of the myeloid sublineage cells. But compared to the syngeneic models, their immune system is intact because it's immune incompetent. So it has all these subtypes of immune cells in the mice, but it's mouse. It's not human. So that's the balance between these two type of models. Yeah. So if you look for like efficacy or biology parts, I would choose the syngeneic models.
Understood. So I really think it depends on the application and what we are looking to do,
Yes.
whether we are testing drug or we are understanding a bit more of the disease mechanism. So, thank you very much for the answer. We have a few other questions coming in actually. So looking at some of the field combination trials, what lessons do you think we've learned in terms of preclinical model selection and maybe biomarker strategy that could help improve, translating these novel drug development targets into the clinic?
So the lessons, the lessons we learned is there's a species differences. Mouse is different from human. In terms of the tumors, because the real tumor in the human, they are quite heterogeneous. But in the mouse, you use a cell line to generate quite a mouse cell line to generate the tumor, but the tumor texture is very homogeneous. They're not heterogeneous. You know what I mean? And also the mouse stroma microenvironment, it doesn't like recapitulate the human tumor microenvironment because the human tumor microenvironment is more complex. So I think this is like a differences between the real world and the the mouse models. So if you want
No,
to like overcome this part, you may think about use the PDX model in the human system reconstituted might be more mimic the real tumor in the clinic. Yeah.
and I think the goal will be to ultimately de-risk the entire process to be the way they are a lot more confident. So I think the use of precision models, genetically engineered models will help in that aspect. So thank you very much for that answer.
Yeah,
So
sometimes it may be like overestimate the efficacy in the mouse model compared to the clinic. Yeah.
Mhm. No, absolutely. So given the importance of the tumor microenvironment composition in PD-1 resistant, how does Gen Pharmatech validate the immunosuppressive or cold tumor phenotypes in its models?
So we have a variety of ways to characterize the immune cells inside the tumor. We either can cut off the tumor and make them become like single cells to do the flow cytometry to characterize the different subtypes of the immune cells, such as CD8, CD4, Treg, or NK, and all the other like myeloid lineages by flow cytometry, or we can just do the tumor, make them like the PPFE, we fix them and to do the IHC, which will remain the intact composition of the tumor, and we'll slide them to stain them with different markers. We can do IHC or we can do immunofluorescence with multiple markers together and to look at the composition, the infiltration of the immune cells in the tumor. That's another way to look at the infiltrate lymphocytes.
No, thank you. Yeah, thank you very much. So I think we are a CRO, so I think we definitely offer custom. It does seem that we offer custom solutions to our clients. So if you have a specific assay you like to perform, we'll be happy to perform it for you. So we have some questions are coming in a lot more. One question is, does Gen Pharmatech work on patient PBMCs who are treated with anti-PD-1? So maybe that's the question.
So far, all the PBMCs are from healthy donors.
Okay.
Yeah.
I see.
So yes.
Right.
But if you want to like collaboration with patients, PBMC, we can talk about that later for collaboration.
Absolutely. Yeah, and feel free to reach out to us, or we will reach out to you, with regards to further discussion. In another question, he's asking about I'm guessing, I'm guessing any other syngeneic models that we can recommend for triple negative breast cancer, apart from EO771.
That's a quite a rare like cell line, but I haven't seen this that we generate this model in our cell list. But what do we have for triple negative? We have the MDA231, we have the MDMB468. I'm not sure if we have this EO771.
So are there any other recommendations? Are you aware of any other syngeneic models that could replace EO771? I'm guessing that's the question.
We have a look.
Um,
We can have a look of, and if the
yeah.
I think the guest is interested in the mouse tumor cell line, right? It's not human, the mouse breast cancer cell line. I can't remember at the top of my head, but we can definitely look at our cell list if we have any other than this in the mouse breast cancer tumor.
Mm-hmm.
Mm-hmm.
Yeah, absolutely.
Yeah, you can leave the email. We'll email you back about if we have.
Absolutely. Definitely, and we can also reach out to you. So I guess another question is maybe not on specifically PD-1, but mouse model for vaccine studies. Or I think do you have any insights on that, Dr. Lee?
Yeah, we are doing some like vaccines with some clients. We can do these vaccine studies in some of our facilities. So if you want to talk about something in more detail, we are happy to talk about that.
Definitely.
Yes,
So
we also have this MMTV, the polyoma virus, I think, for the spontaneous breast cancer model. I think we have that as well.
Yes.
Yeah.
Yep, we do have that. That's correct.
Yeah.
They develop spontaneous memory tumors. So maybe that could be also may not be maybe to the question that the other audience member asked about alternative for breast cancer model. And I think she elaborated, the audience member elaborated not just in TNBC, but in breast cancer in general. Perhaps the AMTV mice spontaneous memory tumor models could be used in that respect as well. If you like, we'll be happy to reach out to you, with additional clarification. Yeah. So we're happy to further discuss this. So I think that's yeah. So thank you again. So I think it's a good time to maybe draw a close to this webinar today. I'd like to thank you again, Dr. Lee, for your really thoughtful and insightful presentation, and thank you for all our audience today for joining us and for the live discussion. So if you'd like to learn more about Gen PharmTech's immuno-oncology platforms that include validated models for evaluating checkpoint inhibitors and combination therapies, please feel free to visit our website or reach out to our team directly. A recording of today's webinar will be sent out shortly, so please stay tuned. Thank you again, and we look forward to seeing you at our next event. Take care.
Thank you very much. Thank you. Have a good day.
Bye-bye.
Thank you. Bye-bye.